since I see a lot of friendly faces, but I will go ahead and just introduce myself very briefly. So my name is Darren Daly. So I'm the principal statistician for our clinical research facility, and also a senior lecturer in our School of Public Health. So as a background is in epidemiology and biostatistics. And so a big part of the work that we do in terms of the clinical research facility is really around helping to ensure that the people's studies that we support in terms of patient-focused research and clinical research are rigorous uh, in particular with respect to their study designs, how data are collected, how data are analyzed and reported. And so me as someone who's involved with this subcommittee on research integrity now within the college, I kind of view research integrity, the thing that I guess stands out to me again is this focus on the importance of rigor. If what we're doing in science isn't rigorous, then really what's the point at all? So I find this framing very, very appealing for helping me to justify my job. You know, this is what I'd like to do, is what I like to help other people do, is to ensure their studies are rigorous. And the reason that I wanted to kind of talk to people today was something that really jumped out at me in the fall, and it was following the update to the Helsinki Declaration, which of course is our, kind of North Star for the conduct of clinical research for human research more broadly. Obviously, many of us have been involved in many papers that will say that we followed, the Declaration of Helsinki, and that kind of guided our ethics and the rest of it. And the thing that really jumped out at me, though, with this most recent update, was it really emphasized a number of things I think that speak to, those of us interested in research integrity would ourselves be interested in. A lot of stuff around transparency and openness, but also this really strong emphasis on scientifically sound and rigorous designs and this term research waste. Now, that is a term that I've talked about many, many times before, and I'm going to talk about it again. So if you have talked about research waste with me before, I apologize. It's going to be beating the dead horse. But experience also tells me that lots of people still haven't been exposed to this kind of idea of what research waste might be. And so the basic concept really goes back to primarily this paper by Chalmers and Glausio in 2009. They published in The Lancet. And, one, we're talking about clinical research primarily. We're talking about the research that tends to happen, or a lot of the research that happens in our college, which is very much on the applied side of things. And so the first focus in terms of the research waste framework is just to think about ensuring that questions that we are asking and we are using research to provide answers to are the ones that are most important and most important to the people that they should matter the most to: policymakers, patients, clinicians, banking decisions, and so on. And I know that everybody in this call, I'm supremely confident that every one of you are here doing the job you do because you want the research that you conduct to actually, God forbid, make the world a better place. And so this is why we have things like priority setting partnerships. This is why we have a lot more input from PPI, trying to help us to, ask better questions that matter the most for the people that we care about the most. Another big source of research waste is, this is research that isn't going to inform decision making, is that a lot of the research just isn't accessible. And, so obviously there's a lot of research that just doesn't need to get published at all. And a lot of research that when it does get published, people just can't get access to it. And so again, if you've done this piece of research, you think it's important, you've got a good question that's relevant for policymakers and decision makers, but nobody can actually get access to the information, then obviously that is a wasted opportunity. And, we have a lot of biases in how we publish stuff, that's for sure, and certainly true in medicine. And, one of these, for example, one of my favorite examples is looking at what happens when we increase transparency around outcomes. So essentially that plot is showing you effects from large clinical trials of pharmaceutical and dietary supplement interventions. And these are the results based on the stated primary analyses. And if you could just mute yourself, I can't switch back and forth between the presentation and the people tab. So if you're not muted, please mute. So anyway, this just shows these effects for the stated primary outcomes in each of the studies leading up to 2000. In the year 2000, there was a policy change. It's required people to register their primary outcomes on clinicaltrials.gov, which really encouraged people to not outcome switch, was a concern that people were having. And then if you look at the outcomes following that policy change, all of a sudden, all the outcomes are now mostly null. So it was, everything miraculously worked before that intervention around transparency. And now since then, it's obviously a very different story. So that accessibility and selective publication of findings is a very important threat and generator of research waste. And then another point is that, we all write papers, we all read papers, and certainly many of you have conducted systematic reviews and other kind of syntheses. And one of the first things you learn when you do that kind of work is you probably find yourself cursing the various authors because they're all just out there doing something very differently. So again, there are interventions out there, so to speak, to help us do a better job of reporting things clearly, reporting things consistently, so that the research we produce can be used by policymakers, decision makers, and whatnot. And so many of you will have been exposed to the various reporting guidelines that now exist in some kind of effort to standardize, again, the things that we're saying. And certainly in clinical trials world, we would be using Consort to guide, us as our primary source of direction for what we'd be sharing and writing. And I'm sure, like I said, many of you have used these as well. So that brings us to the big blank, which is what we're going to talk about a little bit more today. And of course, this is within my wheelhouse, which is research waste due to appropriate design and methods. And again, I'll show examples as we go through this about the kind of stuff we're talking about. But for the moment, you can please take my word that lots of stuff gets published. It gets through peer review. And overall, it's not hard to find examples of published studies, even in so-called high impact or very prestigious journals, where the methods in terms of the analysis and the design and the analysis of the data are deeply, deeply flawed. And again, that goes back to 2009. The Lancet did a full series on research waste a few years later, I think 2013. But very importantly, this research waste story, so to speak, that's now found its way into the Helsinki Declaration. We can also find it in the HSC research strategy. Here they borrow this figure from the NHS research forum framework for adding value in research. And again, this shows up in the HSC research strategy. So with a very welcome addition of efficient research regulation and delivery, which I know a lot of you would be very interested in, all those challenges about actually getting research done is another source of research waste that's important for us to deal with. But this is a message that's out there now, and something I think that if you weren't aware of it now, you are now aware of it. And that comes with certain responsibilities to make sure that you're avoiding research waste. There's, of course, a history kind of leading up to that point. We're not going to go very, very deeply into it. I think in terms of medicine and health, one of the foundational papers is this one, was published in 2012. And essentially, this was a couple of scientists from Amgen who tried to replicate a number of preclinical studies that they had, formed the basis for drug discovery efforts within Amgen. And they just weren't able to replicate the results from those studies whatsoever. And obviously, this raised a lot of concerns about, why couldn't we produce these results, and what does it say about the quality of the research that we are relying on to make decisions. Again, without going into, a kind of in-depth discussion of how research can go wrong, you don't have to take my word for it. I think these are quotes from the respective editors of the New England Journal of Medicine for a very long time in the Lancet. And, both of them expressing very serious concerns about the quality of the research that appears in, the journals that they publish. I think these are very brave statements because in some ways, as the positions they held, they're somewhat complicit in exactly what they're saying here, but very concerning. And again, going back a little further, um, we can talk about the root cause of research waste, really. And for those of us, certainly for us statisticians, we often point to this really important paper in the BMJ called The Scandal of Medical Research. And this was written by Doug Altman, who is head of statistics for the BMJ. And all this paper does, it's really just like 3 or 4 paragraphs. Doug Altman just makes the point that if a surgeon was doing surgery not using the best possible methods, it would be a scandal. But all the time we see evidence of research being published, research that goes into guidelines and helps us understand, how to practice medicine. We see that research being done all the time without the best methods, and yet it's not a scandal. And this is back in 1994, and really what Doug did, and he certainly wasn't the only one, is Doug pointed directly to really it was about publication pressure that was the major driver of this. In other words, we were doing research not because we needed to get answers to important questions that were going to help patients in society, but we were mainly writing papers because we needed to publish them so we could get promoted and advanced and be seen to be doing research. Doug leads off with this great quote, we need less research, better research, and research done for the right reasons. But I can assure you that suggesting we might do less research is not a winning strategy in any university in 2025. And I would just highlight that if we were to, if you look at our current university strategy, which has got a bunch of great stuff in it, of the kind of metrics it talks about, one of them is, increasing the number of scholarly outputs across all publication types by 10% per year, which is a doubling time of 7 years. So it's basically setting a goal that we will double the number of scholarly outputs that we produce as a university in 7 years' time. And I don't think it's that hard to understand the consequences if we were to do that, of what it actually means for quality, and particularly in a really unfavorable funding environment, which is, of course, not of our making, on how exactly we're supposed to accomplish this. So moving on to the research waste that kind of is more relevant to the things certainly that I'm interested in our efforts in the CRF and beyond. Really thinking about what you can do in terms of your data, how you approach your analysis of your data, how you approach study design, and statistics and interpretation. So I'm just going to go through basically just highlighting some of the stuff we do, some of the stuff that you might be able to adopt to kind of strengthen your own research processes, particularly around data and analysis. And then, after we get done with that, happy to take questions and discuss. So thinking of data, I'm someone who receives a lot of data sets, and I can surely attest that every one of them is uniquely horrible in their own special way, unless they aren't. And there's no shortage of examples of poorly collected data and the rest of it. No shortage. It's not hard to understand if the data aren't correct. Then obviously everything that comes from them it's a poisoned well. One of the kind of more interesting examples, is that people were using Excel spreadsheets to record gene expression data, and Excel was making changes to the data they were entering. And anyone who's used Excel has probably experienced that before. And those changes were being propagated through many papers. So essentially all of these data were in error because of just using the wrong tool for the job, essentially. Certainly one of the things that we've been developing, certainly led out by my colleague Brendan, also with Jerry Deasy and Infant and Research IT within the university, is wider deployment of structured electronic data capture with high quality databases, with high quality systems in place to ensure that data are collected accurately, that we can track who's entering data. We can go back and raise queries and correct data from source information when we need to. And, these are two options that we particularly use: Castor for really for larger trials, and REDCap for smaller studies or studies with lower budgets and less regulatory needs. But ultimately, there's lots of options out there for doing a better job collecting data. I think, than many people would even be aware of. But also the reality is that, many of you are going to be, essentially constrained to use kind of bog standard spreadsheet software like Google Sheets or Excel. And if you're doing that, it's really contingent on you to get the training you need to do that the right way. And again, I know from very personal experience of someone, again, who received lots of data sets, that people aren't getting this training. And again, if you are, speaking to PIs, asking PIs, like, what processes do you have in place to ensure that, the people on your research team are doing things the right way and the rest of it, the answers you'll get to that question are highly variable. And I think it's very risky, really, to kind of rest back and be assured that what's happening with the data, if you aren't the one doing it, that it's being done the right way. I think people are very brave to not have better systems in place. In terms of, there's the quality of the data, and there's also this important need to understand the provenance of the data. And there's certainly a lot more interest in this and a lot more scrutiny of this. So here's an example from a few years ago where the editor-in-chief of Molecular Brain had 41 editorial decisions they were facing, where they needed to see some actual data from the study teams before they could make a decision. And out of those 41 decisions, about half of them just withdrew without providing the raw data. So here they were, they'd been reviewed, they'd gone through whatever process that was. I'm sure it was months and months. And here's the editor-in-chief saying, we're happy to consider this, but we need to see the data. And 21 out of the 41 just didn't even bother to provide it. And then of the 20 that did, the editor-in-chief rejected 19 of the 20 for being insufficient. And this kind of scrutiny is on the way. And if you're not prepared for it, I think people are going to find themselves in very uncomfortable situations. This is what underpins kind of an interest now in fair data stewardship, not just about the quality of the data, that's important too, but also making sure the data are available. So they can not just be reused, but they stand in as evidence of the quality of the study itself, or even that the study took place. So the HRB has obviously been very proactive in this space and have the European funders in making sure that FAIR is an important part. The university has been very proactive about this as well, and building an importance around open research and FAIR, which would be kind of fall under that umbrella as an important part of what we do here in Cork, and that's very welcome. And then just to shout out for people who are doing research data management and stewardship, we do have a national network now that was funded by the National Open Research Forum. This is a national project, but it's been led out by our own Aoife Coffey in the library. And so we're developing training materials and things like that, really to help people get up to speed with managing their own data. Because the reality is that most people, they're not going to be involved in studies where they have external data management supports. And so they are going to have to figure this stuff out for themselves. So we definitely need to be providing that training, and that's part of what SONRI does. So the data are one thing, but let's say that we wind up at the end of the study, we've got a great data set, we've done everything we need to do in order to collect the data accurately. There is still the process of analyzing those data. So, there's the statistics and the design, but then there's the actual, keystrokes required to produce the results from the data. And certainly this is an area where I had my eyes opened when I moved into my role within the clinical research facility, in that I didn't pay that much attention to how I was analyzing the data. I just produced results. I assumed they were what I wanted from the first place, and I reported those and moved on. We are now seeing a lot more kind of evidence of what happens when that's the approach you take. Here's an example from 2023. This was a JAMA pediatrics study about COVID in children. And I just highlighted all the bits where it said coded in the retraction. Essentially, the entire retraction was based on just a large number of coding errors. So this is just, instead of, labeling this variable this way, they've labeled it the opposite way or whatever. And again, like, kudos to these authors, for one, being able to catch the errors, and two, raising their hand to say they were there. But these are very easy mistakes to make. And I tell people that my biggest enemy is my two big fat thumbs, right there. You have to type, you have to do things to analyze data, and we are all error prone. We can all make mistakes. And so it's really critically important that we have processes to ensure that when we make mistakes, as we inevitably will, that we're able to identify, catch, and correct them, or have a high probability of doing that. Another very stark example of exactly this is if you look at the plot on the left. This is comparing intervention to usual care in a study that initially people said, oh, well, this intervention is hugely beneficial for patients. So we need to make sure that that's something that goes forward. Thankfully, the error was caught, which was that they had flipped the labels on the two treatment arms in the process of doing the analysis. And actually, the intervention was substantially harmful to patients. Again, this was done. This is a paper published in JAMA, not JAMA, whatever, just the JAMA. And this was done out of the Johns Hopkins Biostatistics Center. So not to throw shade at them, the point is that these are people who really, really, really know what they're doing, and they're capable of making errors. So I know we are, and I know I am. But again, they were able to catch these errors. So for you, as someone who, if you find yourself as someone who needs to analyze data as part of your job, and that's not everybody, but it's certainly many of you, we really want to encourage people to take a proactive approach to improving the quality of how they do this. And this isn't to criticize SPS particularly, or its use. People often, they just use the software they were trained in at some point, and that makes a lot of sense. But what we really want to do is to ensure that people are moving away from a kind of point and click style of analyzing data, where instead people are scripting exactly what they're doing. We use R, which is a statistical programming language, and then we use essentially an interface called RStudio that makes it much more user-friendly to work with. And, for a lot of people, showing them a wall of code, can be quite intimidating, but that's just because you're not familiar with it, right? And when, when you're a smart, accomplished person and someone throws something at you haven't done before, it's going to generate a certain amount of discomfort. And if someone came to me and gun to my head said, Darren, you have to do this analysis in SPSS by tomorrow, I would experience an awful lot of stress because it's not what I use. I would have to learn a whole new thing and very quickly. But hopefully the point of value of a scripted analysis is clear. When we all learned about science in grade school, you write down what you do in painstaking detail, and that way you can replicate exactly what you've done. And that's all the scripting does. And the only learning there is having to, learn what the specific things you need to write down in order to accomplish your aims in the analysis. And once you've learned that, then you've got it. But also doing this, it's not just that you can now find your errors and you can have processes in place to help you do that. You get lots of knock-on benefits in terms of efficiency of your work. You can start to recycle the code that you've used. Certainly things that you've used for one project, you'll almost certainly use them for others. So in a lot of ways, it's not just about the quality, it's about making you more efficient. And also making you more efficient gains you time. And probably the biggest driver of research waste, I don't think I'll get a lot of disagreement on this, is the biggest threat to research integrity is the enormous time pressures most of us face. And so if you can cut hours out of your schedule, by making the way you do things more efficient, that's obviously going to be good for the quality of the work you do. Another nice trick with the way we do things is, again, we can embed our analysis work with the reporting. So we're not cutting and pasting results from one source to another. We're trying to develop reports, or we do develop reports where, essentially human readable text that you've written is embedded with the machine code, and that gets produced and embedded into the same document, which again is just a very nice way of doing things very efficiently. And if this isn't a thing you're doing or a way you understand of working, Again, we have training for this. So there's my colleague Brendan. He's been doing a postgraduate module in reproducible research with R for a number of years now, and I think has put through 250 or 300 postgraduate students at this point. And again, this is something that you need to engage in. If it's not how you're working, it's something we very strongly suggest. But at the very least, no more point and click. You've got to get over to a scripted way of doing things. And again, if you're a PI and you're relying on your teams to be producing, analyses out of the data, I would want to have, processes in place to make sure that people are doing that the way they should be, and training and all the rest of it. Because ultimately, we do kind of have this thing, we know it's about our big ideas and we don't want to tamper with people's creativity. But, again, go back to that research integrity statement at the beginning. It's really is about professionalism and rigor. And that includes this too. And if you're working, in a lab, obviously you have very clear procedures. They're not loosey-goosey. You're supposed to do things a very certain way. That's just a part of science. And analyzing your data and collecting data is really no different. Okay, so we'll finish up talking a little bit more then about statistics and design. And so just the best personal example I have was I played a very small role in this, what was called at the time a living systematic review, and published in the BMJ. And this was of COVID risk prediction models. So models that people were using to either diagnose COVID or to make prognosis from COVID diagnoses to help maybe guide treatment and care. And so this project brought together a pretty large team, and we started reviewing all of these models. Because essentially, you will remember at the start of the pandemic, people left, right, and center wanted to come up with ways that they could help. There's a lot of data out into the world. There's a lot of algorithms for producing or processing those data and turning them into predictions. And certainly lots of people went to that task. And so we wanted to review those as they were being written and evaluate their quality. And again, it was a living review, so it got updated a few times. And I think we wound up with over 350 models that wound up being reviewed. When you went through and did the reviews and kind of summarized everything, again, this is people, at a time of crisis, people trying to put their best foot forward. The overall findings of the review were really pretty grim. And if you kind of considered, never take these risk of bias tools, always take them with a grain of salt. They're heuristics at best. But as someone who reviewed many of these studies that fed into this, you see the big red bar here around analysis, that this was an area where people were making choices about how they were analyzing data that a large group of experts would have, questioned. And, that was the story of the review, essentially that the vast majority of the studies that went into that review were not of a good quality and weren't going to move the needle clinically in any way whatsoever. They were research waste. And I can also assure you that a vast majority of those studies that went into the review would have, when they were published, been accompanied by a glowing university press release extolling the wonderful work that had been done. But it's just simply not the truth. In terms of regulated clinical trials, we have some, kind of regulatory backup for the importance of certainly for statisticians. So, if you are involved in regulated clinical trials, good clinical practice is very clear that you need to have a trial statistician involved in your study. The little bit at the top here, this is an excerpt again from Helsinki, so they're not quite as direct, but of course they're talking about a much broader class of clinical research, not just regulated trials. But again, highlighting the fact that research involving human participants really needs to be conducted by people with the appropriate training. Science is a team effort, and certainly clinical research is a team effort. And it requires a variety of expertises to do things the right way, and statistics is just one of those. Of course, the reality is that most of us do have to be our own statisticians. So people who are working with quantitative data, you've got to be the methods expert, at least to some kind of a good standard. And for people who do, qualitative and other forms of research, I'm sure it's exactly true for those areas as well. And one thing I'm fairly confident of, having heard lots of feedback from people over the years, and certainly lots of, reports from other places, we're not unique in this regard, is that graduate students and earlier career scientists, they feel a need, right? They've gotten some training, they recognize they need more, and while they're training and while they're becoming more experienced, they need access to expertise and support in order to ensure that the studies they're doing are very high quality in terms of statistics and analysis and design. Uh, Here's an example, a very straightforward type of paper to write. This was one that went into the literature and just picked out 100 studies at random that reported doing their sample size with G-power. There's nothing wrong with doing a sample size with G-power, of course. It's just that it's an easy thing to search for. And essentially, the vast majority of these did it the wrong way, or they didn't report it clearly enough to where it could be replicated. And so, that's stuff that goes into the very design of the studies. And an underpowered study, or that's by definition going to generate research waste. If you get a null result, you don't know if it's because there was no effect or if it's just a function of design. So again, this is kind of pure research waste, and it's very easy to demonstrate that there's a lot of need here in the clinical research and medical research community to improve how people go about things. So I thought I would just finish up with kind of two more kind of explicit examples. If it was kind of, Darren, what are things that people should avoid doing? Very easy things to do from a statistical point of view or a design point of view. And one of them at the top of my list is always going to be that people frequently they categorize variables they should. And so we're measuring things all the time, and a lot of those things are very continuous in nature, of course. And a continuous variable or a continuous set of measurements, it contains a lot of information, right? It contains information about order and distance between different values. But frequently people want to categorize things. So you essentially throw all that information away, right? So if we were to categorize this number line here at the 5, now we're treating someone who's a 4 basically very differently than if they were a 6, but the same as if they were a -10, right? This is a loss of information. And I think what often people don't appreciate is that loss of information has really significant consequences for essentially the power of your study or the accuracy or precision of your estimates. And so just as an example, you could think about a study with a continuous outcome. So let's say we had 2 groups, and we could do these density plots to represent the distribution of this outcome within those 2 groups. And you can see the distributions overlap quite a lot, but we put in, a line for the mean value in each of these groups. And so we might want to devise a study and a statistical test to tell us something about this difference. And so if we were to do that based on a difference in means, we could, look at the power we would have to go off and try to detect that level of difference. So a standardized difference in means of -0.5. And we could look at the sample size you'd need in each group to get some level of power. And essentially, we get up here into the area where people are often trying to power their studies of 0.8, 0.9, around 50 to 100 patients within each group. So if someone came to me with this kind of design, this is maybe the result I would give them for their study planning. If instead, though, you were to define your outcome not as the continuous outcome and look for a difference in means, but if you were just to define your outcome as, say, people at or below, some cut point. And here I've made it a cut point that's, 10% of the control group would have a greater value of that. That same cut point, so this bit of the distribution here is 10% of those patients in the blue group. It's 3.8% of patients in the active group. So again, it's reflecting a similar idea that the outcomes in the active group are lower than outcomes in the control group. It's the same data, the same information, except here we're categorizing. And if I were to then do the same power analysis based on that, that's what the power curve looks like. So we're going out to 300 patients in each group in order to have the same power to detect what is effectively a same difference in the underlying data. But because we've categorized things, we've thrown away so much information, that propagates into the power of the study overall. And if you were to think about how hard it is for those of you who have, recruited human studies, and how many studies fail because they don't manage to recruit the number they set out to recruit, that's a huge generator of research waste. That's a lot of studies that get off on the wrong foot from the outset. And had they been, better designed and more efficiently designed, maybe that would have been the study, that would have found the thing that actually might help patients. And I often think people sleep a little bit. We're very concerned about false positives. We're very concerned about saying a thing does something beneficial when it really doesn't, and we should be. But missing beneficial effects is obviously hugely important in clinical research, because who knows if that idea even will be investigated again. Maybe there's some benefit there that gets lost forever, and maybe just as a function of kind of a substandard design of the study. Finally, I'll finish on this example. It's a fairly recent one, but it's something that I see people do all the time. They will do a very sensible thing in a comparative study trying to understand the benefit of some treatment, and they will run a comparative study, right? We have control groups for a reason. If I take a pill and my symptoms resolve, it's very hard for me to say whether, in most situations, whether the pill actually did the thing or not. So we run randomized comparative studies. We randomly divide people into two groups in the most basics, example. We give one the treatment, we give someone else standard of care, we use a placebo for blinding, and then we make sure that all of our estimates of the benefit of that intervention are between groups. And so here's an example from a paper that was published in the American Journal of Gastroenterology. And the table is the number of outcomes down the left. And then it gives us p-values for the difference in outcomes between these two groups. One group got the fancy new thing and one group got the standard of care thing. And all the p-values are very large and nowhere near our kind of usual cutoffs for, piquing our interest and whatnot. And so from that perspective, and really the only perspective that matters, this is a phenomenally null trial. There's no difference in, there's no anticipated benefit in the new thing compared to what they do already. Unfortunately, what these authors then did, and this happens all the time, is they then decided to focus on the within group change. And again, within the two groups, things did get better for both groups. But again, that means that one wasn't any better than the other. But somehow the authors were able to find some differences in significance within the within group changes. So they then argued, even though the between group difference was quote unquote non-significant, we found a significant improvement in this group, in this thing, and not in the other group. And that's how they made their inference that this was a beneficial intervention. They didn't stop there. This is how they actually said it in the paper. So, you read, and again, this is the American Journal of Gastroenterology. This is a real editorial board. This was reviewed and the rest of it. So despite the phenomenally null findings of the study, they managed to get this conclusion into the abstract that this thing that they were doing is hugely beneficial. It's a valuable strategy. It's exactly, what we should be doing. And, it's not that hard to to see the disconnect between the actual data and whatnot. We wrote a letter, and so you can read this. Don't read it right now, please. But you can go find, we wrote this, and they were very kind enough to publish this letter in response, essentially outlining the points I've tried to make here today. They were very kind to let us keep the title of the letter, Study Design 101, to make this point that estimation of treatment effects should be based on between-arm comparisons. And importantly, too, you can go to the author's LinkedIn page, and they will tell you all about their groundbreaking multi-center study and how the thing that they're selling to people is very, very beneficial. And the American Journal of Gastroenterology, essentially, by publishing the paper, despite all of its flaws, gives them the opportunity to make these claims, even though they fall down under any scrutiny whatsoever. So doing stats and design the right way is, of course, very important. And being honest with ourselves is too. But that's maybe a topic for another day. A few quick things just to say that, coming back to this point about how you get support for statistics and the rest of it, it's not an easy problem to solve. And the reality is that if your day-to-day job, as a scientist involves analyzing some data, it really is contingent on you to take that part of your scientific work seriously. I people maybe heard this before. Like, I have a lot of sympathy for clinician researchers that are already doing another job, and to become an expert statistician is certainly a lot to ask, and I think that's a lot to ask of anybody who's not focused on that full time, obviously. But there's plenty of people who don't have any clinical responsibilities. Their job is full-time scientists. Their job is to design studies and report results and analyze data and the rest of it. And if that's the position you're in and you're feeling uncomfortable with statistics, then feel free to reach out, number one. But there are places you can go to kind of improve your understanding, and it doesn't have to feel like a terrible thing. One of my favorites is this long series of statistics notes published in the BMJ. You could do much worse than to just pick one of these papers a week. They're all very short, and careers tend to hopefully long. Pick a paper a week and go through it and learn some stuff. And then I've written some stuff here that you're welcome to go find. That's got a whole bunch of other links about how we can all improve our own statistical kind of training. And with that, I will shut up and happily take any questions.